Αγαπητή Μαρίνα, τα θερμά μας συγχαρητήρια. Ευχόμαστε μεγαλύτερες και περισσότερες επιτυχίες. Θα σε παρακαλέσω, όπως έρθεις, να παραλάβεις το βραβείο σου, που είναι μια πλακέτσα και ένα πιστοποιητικό και ένα βραβείο χρηματικό, όπου θα σου μεταφερθεί ηλεκτρονικά πριν μας παρουσιάσεις την έρευνα σου.
Hi. It is with great honor I received the Junior Scientist Award in memory of Professor Panos Ioannou, an internationally acknowledged cell biologist and neuroscientist who pioneered in research on thalassemia and led the field of cell and gene therapy. I am humbled to be awarded in the memory of a leading scientist who made a difference while maintaining solid work and science ethics. Today, I'm going to present you a translatable RNAi gene therapy approach for CMT1A neuropathy and briefly navigate you through the route towards its application in the clinic. This work I'm presenting today was published on the cover of JCI, is protected by world patent, and had received orphan drug and rare disease designations from FDA. CMT is a large and genetically heterogeneous group of inherited neuropathies that affects almost 2.6 million people worldwide. CMT neuropathies usually have early onset, starting from childhood to adolescence. In most CMT types, progression is very slow and disabilities accumulate gradually during life without being life-threatening. Among others, affected individuals are typically characterized by distal muscle weakness and atrophy and moderate distal sensory loss. Symptoms may also include muscle pain, hand tremor, cold hand and feet, nerve pain, chronic fatigue, numbness, curled fingers and toes, high urge, and breathing problems. Currently, there is no accepted treatment for CMT neuropathies, and their management remains mostly supportive and symptomatic. CMT1A is the commonest type of CMTs, resulting after the duplication of PMP22 gene. This creates a gene dosage effect that destabilizes the myelin sheath, leading to axonal degeneration in distal and proximal nerves, as well as in the spinal roots. PMP22, the protein which causes CMT1A, is an intrinsic tetraspan glycoprotein primarily produced by myelinating span cells during development. This protein is crucial for span cells' growth and differentiation, myelogenesis, myelin thickness, as well as for the maintenance of PNS axons and myelin. Despite the fact that PMP22 mRNA is ubiquitously expressed in every tissue, PMP22 protein is only found in myelinating span cells, with this suggesting a post-transcriptional regulation of PMP22 mRNA. CMT1A genetic mechanism depends on the gene dosage effects of PMP22. Overexpressed PMP22 has been shown to saturate the proteasomal capacity for degradation, leading to perinuclear and cytoplasmic PMP22 accumulation, decreased overall proteasomal activity, and ER stress. Therefore, our goal was to develop a translatable method to reverse and restore the genetic mechanism of CMT1A in order to ameliorate the phenotype of the disease. To achieve this, we designed, in collaboration with Professor Harper from Nationwide Children's Hospital, multiple novel artificial PMP22 targeting microRNAs that were tested in vitro, and then the most promising was packaged into an AV9 vector. In parallel, we performed a detailed characterization of a CMT1A animal model. Once this was completed, we evaluated the in vivo effects of our microRNA in CMT1A animals at 3 different treatment trials, which led us to the conclusion that our MIR is actually promising to provide therapeutic benefit. At the end, we also examined the possibility of over-silencing effects by testing our microRNA in wild-type mice. Let's start with the design and the in vitro testing of our novel artificial MIRs. Our microRNAs were designed based on the natural MIR30 scaffold. Designing and selection criteria of the most promising microRNA was performed following the criteria listed here in B. All sequences suggested by our algorithm were found to target the 3' UTR of both human and murine PMP22 exon 5. For in vitro testing, HEK cells were co-transfected with either the human or the murine PMP22, along with each of our artificial microRNAs. Cells were then harvested and analyzed by real-time PCR, which suggested MIR871 as the most promising in silencing both human and murine PMP22. We wish to use a microRNA that equally targets both human and murine, as we wanted to have in our equation the potential of overall silencing side effects. Based on these results, the U6-driven MIR871 was packaged into an AAV9 vector along with the CMV EGFP element. U6 and CMV are both ubiquitous promoters. For control, we used an AAV9 vector, which was expressing a microRNA targeting the LAGZ gene, a gene which is not expressed in our mice. C61 head animals is the model that best resembles CMT1A in terms of both the genetic cause and the resulting phenotype. Despite the fact that C61 animals were first described more than 20 years ago, no one except us now had provided a detailed characterization of the model. Therefore, for better interpretation of upcoming gene therapy effects, we consider it fundamental to first gain insights on the C61 head baseline. We started by performing timeline behavioral testing in CMT1A animals in comparison to wild-type mice. For this purpose, CMT1A and wild-type mice were monitored every 2 months, starting from 2 months until 10 months of age. This analysis included rotarod at 2 different speeds, grip strength, hang test, and hind limbs opening evaluation. For all the tests employed, performance of CMT1A mice was significantly worse than wild-type and had showed progressive worsening with age. We have continued with electrophysiological analysis of CMT1A animals, which showed impaired motor nerve conduction velocities as early as 2 months of age and reduced CMAP scores from 4 months of age and onwards. We also analyzed the levels of 2 axonal degeneration circulating biomarkers, NFL and GDF15. NFL was found elevated in CMT1A animals starting from 6 months of age and onwards, while GDF15 showed progressive worsening with age. We completed our line characterization with morphometric analysis on semi-thin sections of lumbar motor roots and femoral motor nerves. Lumbar roots showed a progressive demyelination, which was characterized by thinly myelinated and demyelinated fibers as early as 2 months of age, and by onion bar formations, which became apparent from 6 months of age and onwards. A very similar pattern was also observed in femoral motor nerves. Since we now had a very clear understanding of our CMT1A animal model, we proceeded with intrathecally injecting our AAV9 MIR vector in adult CMT1A animals that were analyzed 6 weeks post-injection for biodistribution. Remember that along with our artificial microRNA, we also express ubiquitously the EGFP reporter gene. So according to this immunohistochemistry and vector genome copy number analysis data, our vector is concluded to efficiently transduce both PNS and non-PNS tissues, resulting in a widespread biodistribution. Knowing that our vector now reaches the tissue of interest, we proceeded with examining its silencing effects by real-time PCR and Western blot analysis at 6 weeks post-injection. Here we see that both at the RNA and the protein level, our MIR871 silences human and murine PMP22 in roots, sciatic, and femoral nerves, with this being followed by elevated levels of other myelin-related genes and proteins. So until now, we have shown how we designed our artificial microRNA, how we tested that in vitro, and how we packaged the most promising in an AAV9 vector. We have also shared with you a detailed characterization of the C61 head mice, and we have also confirmed that intrathecal injection of an AAV9 MIR871 is an efficient method to silence PMP22 genes in vivo. These milestones allow us to proceed and address the question whether AAV9 MIR871 is actually efficient enough to provide therapeutic benefit to CMT1A animals. To answer this question, we designed a proof of concept experiment, which was consisted by 3 treatment trials: the early, the extended early, and the late treatment. For the early treatment, 2-month-old CMT1A mice were intrathecally injected with the AAV9 MIR871 vector or the MIR-LAGZ vector and were monitored every 2 months by behavioral testing until 6 months of age. At the final time point, we also analyzed our treatment group with vector genome copy number analysis, real-time PCR, Western blot. We performed electrophysiology. We counted the circulating levels of NFL and GDF15. We performed morphometry and immunohistochemistry analysis on their tissues. Vector genome copy number analysis and immunohistochemistry showed stable and efficient transduction of both PNS and non-PNS tissues at 4 months post a single injection. We also confirmed long-lasting, robust silencing on both human and murine PMP22 at the mRNA and the protein level of roots, sciatic, and femoral nerves, with this again being followed by increased level of other myelin-related genes and proteins. We then examine how these long-lasting silencing effects actually impact the phenotype of our model. In blue, we see the miR-871 treated animals. In orange, the MOC treated animals. In pink, we have the non-injected CMT1A animals, and in black, the wild type. At every time point of rotarod, grip strength, hang test, and hind lips testing, miR-871 treated animals performed much better than control-treated animals, with their scores actually reaching wild type levels. At the right, we see the impressive improvement of hind lips opening after treatment. Electrophysiology analysis was in line with behavioral testing, suggesting a total restoration of motor nerve conduction velocities and a significant improvement of CMAP scores. Importantly, the levels of prior elevated NFL and GDF15 axonal degeneration blood biomarkers were significantly reduced at 4 months after early treatment. We also performed morphometric analysis of myelination in semi-thin sections of lumbar roots and femoral motor nerves at 6 months old early treated CMT1A animals. In both roots and femoral nerves, the percentage of thinly myelinated and demyelinated fibers was significantly reduced, while spinal roots also showed reduced number of onion bulb formations, whereas in femoral nerves, onion bulb formations were already low at baseline and hence were not altered after treatment. The analysis of our early treatment group was completed with immunofluorescence to evaluate the inflammatory status of lumbar roots, sciatic nerve, and the liver. miR-871 treatment decreased the prior elevated percentage of CT inflammatory markers in roots and sciatic nerve, while our injection didn't cause any permanent inflammatory response in the liver at 4 months post-injection. We further examined the effectiveness of our treatment in later stages of the disease. For this purpose, we injected mice at either 6 months of age for the late treatment group or 2 months for the extended early treatment group and analyzed them at 10 months of age, using the same test as the ones we described for the early treatment group. Vector genome copy number analysis suggested efficient transduction of PNS and non-PNS tissues at both 4 and 8 months post the single injection. As with early treatment, the late treatment with miR-871 resulted in the downregulation of human and murine PMP22, with this being followed by elevated levels of other myelin-related genes and proteins in all roots, sciatic, and femoral nerves. We compared then the motor performance of late treated with extended early treated mice. In light blue, we have the extended early treated mice. In blue, the late treated. In orange, the MOC treated animals. In pink, the baseline of CMT1A. And in black, the wild type animals. Rotarod, grip strength, and hang test analysis showed the CMT1A mice performed similarly with extended early treated mice, resulting to complete restoration of their phenotype. Interestingly, despite the significant improvement of late-treated animals in hind limb and motor nerve conduction performances, only mice of the extended early treated group reached wild-type levels. However, CMAP scores of both these treatment groups did not improve, and similarly, NFL and GDF15 levels remained unchanged. We performed morphometric analysis of myelination in motor roots and femoral motor nerves from 10-month-old treated CMT1A animals that received either the late or the extended early treatment. Both treatments showed decreased number of onion bulb formations in roots and femoral nerves down to wild-type levels. Percentage of thinly myelinated and demyelinated fibers of these PNS tissues were only totally rescued in the early treatment group, As in the early treatment group, we compared our analysis with immunofluorescence to evaluate the inflammatory status of our lumbar, roots, sciatic nerve, and the liver. The late treatment with MIR-871 resulted in decreased percentage of seeding inflammatory markers in roots and sciatic nerve, while once again didn't cause any permanent inflammatory reaction in the liver. Well, so far so good. We have proved that AAV9 MIR-871 is an efficient way to transduce PNS and non-PNS tissues, and our injection can last for up to 8 months post a single injection, providing a therapeutic benefit in CMT1A animals. And our treatment is not causing any permanent inflammatory reactions. Of course, our data suggests that the earlier the intervention, the greater therapeutic benefit will be, but is there a risk of PMP22 over-silencing? To examine this, we tested our AAV9 MIR-871 in wild-type mice, expressing only 2 copies of mutant PMP22. Injected animals were analyzed by Western blot at 6 weeks and 4 months post a single injection. In parallel, an additional wild-type group was injected with either MIR-871 or MIR-LAG-Z at 2 months of age and was monitored every 2 months until 6 months of age, with the same tests as the ones that were performed in the previous treatment groups. We performed RNA analysis, electrophysiology, we counted the plasma NfL levels, and we performed morphometry. Real-time PCR suggested that mutant PMP22 was significantly decreased in lumbar roots, with this being followed with MPZ reduction levels. In sciatic and femoral nerves, the only effect caused after our injection was an enhanced expression of mutant GJB1 gene. Western blot analysis at 6 weeks and 4 months post a single injection showed significant decrease of mutant PMP22, which affected only the MPZ levels of the sciatic nerves. Behavioral analysis suggested a partial functional impairment of wild-type mice after our injection. Electrophysiology tests showed no effect of MIR-871 in nerve conductions, but a significant reduction in CMAP scores. Interestingly, NfL axonal degeneration circulating biomarkers remained unchanged. Seminal sections of root, sciatic, and femoral nerves showed absolutely no change on myelination or morphometry in wild-type mice after injection. So our last recap slide, we have seen that MIR-871 silences PMP22 in wild-type animals, causing partial impairment in behavioral performance, but no effects in the PNS tissues morphology or no signs of an HNPP-like phenotype. This indicates that under normal conditions, mice have a way to actually respond to abnormal silencing. Well, hope that this was not a marathon for you, as I wish to share some more slides after conclusions. So until now, we showed how we designed novel artificial microRNA that when packaged into an AAV9 vector, MIR-871 stably transduces PNS and non-PNS tissues, leading to a stable silencing of PMP22 and improved expression of other myelin-related genes and proteins. We showed that our treatment approach is spherical, provides spherical therapeutic benefit in CMT1A animals, and does not stimulate any permanent inflammatory reactions. Also, the partial phenotype observed in wild-type mice is not severe enough to raise any safety concerns, especially when considering that our treatment only targets humans with overexpression of PMP22. Overall, we are very confident to say that we provide solid preclinical evidence showing that intrathecally delivered AAV9 MIR-871 is a translatable, safe, non-inflammatory gene therapy approach that improves the phenotype of CMT1A animals. So all the work I just shared with you from JCI was performed using only one dose, only one concentration of this AAV9 MIR vector, and that was the 5E11. During the last year, we and Harper's lab partnered with Armatus Bio on a dose escalation study of AAV MIR-871 in mice. During this SRA, we injected CMT1A animals with different doses of AAV9 MIR, including the 5E11, and then analyzed the animals at 6 weeks or 4 months post-injection. At 6 weeks post-injection, tissue lysates were analyzed for vector genome copy number analysis, real-time PCR, digital droplet PCR, and Western blot, while tissue sections were analyzed with the state-of-the-art RNA-scope technology. We also did analysis of CT markers with immunohistochemistry and histopathology analysis. At 4 months post-injection, we analyzed the treatment effects of the different AAV MIR doses with electrophysiology, and we also did morphological analysis in PNS tissue while also assessing the levels of axonal degeneration blood biomarkers. Meanwhile, our collaborators from Nationwide injected different doses of AAV9 MIR in wild-type mice and performed an extensive histopathology analysis. They also employed different human cell lines that were transfected with our MIR871 and analyzed by RNA sequence in order to test the possibility of off-target effects. Well, lucky you, this is just a teaser for our upcoming publication, as we're not going to share any of this data yet. So all this time, we've been doing experiments in mice while our target is actually humans. I think this image can convince you that mice can be a perfect model, but still highly different from humans. Until now, we've been intrathecally injecting, and the most distal part to reach was just 2 centimeters away from the injection point. In humans, the intrathecally injected product should travel about 1 meter from the injection point in order to reach distal nerves. As you can expect, you cannot just jump from mice experiments to human testing. As in our case, the route towards translation starts with in vitro testing. It proceeds with mouse work, and only if mice experiments are promising, you proceed with testing in bigger animals. And if big animals testing does not raise any safety concerns, you then proceed with clinical trials in humans. And according to the clinical trials results, FDA will decide if a product is actually approved to go out in the market. After all this mice data load, I hope I convinced you that we are ready to proceed with big animals, because we did. We intrathecally injected mini pigs with AAV vectors and control pigs with PBS. These animals were sacrificed 6 weeks post-injection and analyzed for AAV neutralizing antibodies in their blood, and their tissues were processed for immunohistochemistry, vector genome copy number analysis, and digital droplet PCR. The overall goal of this study was to test the efficacy of an IT-delivered AAV to actually transduce the PNS tissues of bigger animals, where the distance from the injection point out to distal nerves is much bigger than the 2 centimeters we have in mice. We're also doing body distribution and inflammatory analysis to test the safety of the IT-delivered AAVs. These experiments are now completed, and we are at the stage of data analysis. And as you can expect, things get much busier when moving to NHPs testing. We injected NHPs with an AAV9 vector expressing our MIR871, and we actually tested this vector at 2 different doses and analyzed the animals at either 6 or 12 weeks post-injection. We performed the standard weekly and neurological examinations and collected weekly samples of hematology, serum, coagulation, urine, and we also performed CSF analysis. At the final time point, animals were under ophthalmic examination, electrocardiogram, we performed the nerve conduction velocity test, we counted the levels of AAV9 blood neutralizing antibodies, we did CSF analysis, digital droplet PCR, Western blot, and we're going to perform PNS morphology tests. The overall goal of this experiment was to actually test our therapeutic product in the closest species possible to humans. These experiments will give us for the first time insights about how an AAV9 that is intrathecally delivered in NHPs transduces the PNS tissues. We will analyze for the first time our AAV9 MIR and see how it biodistributes and if it's safe in NHPs. We know that our MIR871 can potentially bind on monkey PMP22 with mismatches. Nevertheless, we don't consider 6 weeks or 12 weeks enough to cause the HNPP phenotype. Once again, NHP experiments are now completed, and we're now at the stage of tissue processing and data analysis. After completing big animals testing, the route towards FDA approval is quite fixed, but not easy, believe me. It starts with submitting a pre-IND letter to the agency, then you submit the full pre-IND package. You meet with the FDA, you get their response, and then you proceed with the IND submission. If everything runs smoothly, you can start your clinical trials and then apply with the biologics and new drug licenses. Once this years-long journey finishes, you'll then hopefully have the FDA approval to get your product out in the market. Still, during CMT treatments translation, there are challenges that scientists should face. As for the majority of neurodegenerative diseases, the earlier the intervention, the greater the therapeutic benefit will be. But someone should bear in mind that CMTs are not life-threatening. So scientists should be super sure and ready to guarantee the safety of their therapeutic product. CMTs are also slow progressive diseases, and there is still a high demand for valid treatment-responsive biomarkers. Coming to the end, or actually close to the end of this presentation, I wish to express my sincere gratitude to my head, Professor Kleopas, who offered me the opportunity to get involved in the CMT1A gene therapy and participate in the journey towards its application in the clinic. I would also like to acknowledge everyone who contributed in the JCI paper, which was funded by CMT Research Foundation. We wish to express our deep appreciation to Professor Harper, who along with Professor Kleopas and myself are the inventors of the patent describing the work I just presented to you. We wish to thank Lindsay from Harper's Lab for being a very important team player for this project. And we wish to acknowledge our valuable sponsor, Armadius Bio, for investing on moving on our therapeutic product from the bench down to the clinic. We are very proud that we actually made it to the JCI cover. We got our work protected by a world patent and gained the FDA designations, and we're actually very optimistic for the future. Here, I want to share with you these pictures. This is us in the last PNS meeting. In these pictures, you see people trying to treat the same disease using different methods. And these people are so happy when they get to meet each other, exchange ideas and knowledge, and truly embrace each other for their achievements. I will be forever grateful to Professor Kleopas for introducing me in this scientific community where the only competitor is CMT progression. Closing, I wish to share a small story with you, and I will do this bilingual. Κλείνοντας, θα ήθελα να μοιραστώ μαζί σας μια ιστορία. This is not the first time I'm standing on this stage. Σήμερα δεν είναι η πρώτη φορά που στέκομαι πάνω σε αυτήν τη σκηνή. The first time was about 20 years ago, when I was 13. Η πρώτη φορά ήταν πριν περίπου 20 χρόνια, όταν εγώ ήμουν 13, don't do the math. Back then, I have every background to fail. Τότε, πριν 20 χρόνια, είχα τις τέλειες προδιαγραφές για να αποτύχω. And today, I'm standing again on this same stage, actually receiving an award. Και σήμερα ξαναστέκομαι πάνω σε αυτήν την ίδια σκηνή για να παραλάβω ένα βραβείο. I consider today a full circle, and I wish to open a new one. Θεωρώ λοιπόν ότι σήμερα κλείνει ένας κύκλος και θα ήθελα να ανοίξω τον επόμενο. That's why I want to dedicate this award to a young lady who is very into science, and I wish to encourage her to keep on moving on. Γι' αυτό λοιπόν θα ήθελα να αφιερώσω αυτό το βραβείο σε μια νεαρή δεσποινίδα που πραγματικά της αρέσει η επιστήμη και θα ήθελα να την ενθαρρύνω να συνεχίσει να προσπαθεί. This award is dedicated to Miss Nicoletta. Αυτό το βραβείο είναι αφιερωμένο λοιπόν στην Νικολέτα, που σήμερα μπήκε για πρώτη φορά σε αυτό το δωμάτιο και είμαι σίγουρη ότι σε λίγα χρόνια θα βρω τον εαυτό μου να κάθεται κάτω σε αυτήν τη σκηνή και να χειροκροτώ εγώ την Νικολέτα.